All right, in this video, we're talking about blood vessels, and I want to relate it to blood flow and blood pressure. It gets a little bit confusing because blood vessels constrict and dilate for really two completely separate reasons, and so we need to try to dissect it and make sense of it all. Okay, the first thing to point out is most of our regulation happens in arteries. So remember, arteries just mean vessels that are carrying blood away from the heart. In the systemic circuit, they're going to be highly oxygenated. In the pulmonary circuit, they're not. So it's not really about oxygen or not. It's about their anatomy. And there are a couple different types. So elastic arteries get their name because they're incredibly stretchy. They have thick walls, lots of elastic tissue. The aorta is the ultimate example of that. And because they're almost entirely elastic tissue, they do not constrict or dilate. So this term vasoconstriction means the smooth muscle of the artery is contracting to squeeze the blood vessel and make it smaller. That is not the same thing as expand and recoil. Expand and recoil means like a rubber band. You can stretch it out and it'll try to snap back. Vasoconstriction, vasodilation is smooth muscle. That's an intentional contracting and relaxing of muscle tissue. So they both involve the blood vessel getting a bit bigger or a bit smaller. But just remember, the expanded recoil, this is just a pressure issue, right? When pressure goes up during systole, when the ventricle is contracting, that pressure stretches out big arteries like the aorta and makes them expand. And then when your pressure goes down during diastole, the elastic fibers snap back. That's what keeps your pressure relatively high. And so when we see a systolic over diastolic of 120 over 80, if you didn't have that stretchy expand and recoil property in those big arteries, this diastolic number would go dangerously low, like almost to zero, right? It's the snapback, the recoil that keeps the blood pressurized and pushing through the body while the ventricle is busy relaxing and refilling. All right, muscular arteries. Muscular does not mean that they lead to muscles, although some of them do. It means they have a lot of smooth muscle in the wall of the artery. These are one of the sets of blood vessels that are active in vasoconstriction, and the idea is they're regulating exactly how much blood you deliver to individual organs. All right, remember, blood flow is represented by Q, and we say Q is proportional to pressure over resistance, right? The most important part of this as far as blood flow goes is vasoconstriction increases resistance. And if you increase resistance, you're going to decrease flow. Think of it exactly like tightening the knob on a faucet at a sink. When you tighten the knob, you're just making the opening at the faucet get smaller so less water can come out. Just like when a blood vessel vasoconstricts, right? The net effect is the lumen gets smaller. Right, so if the blood vessel initially looked like this with a big lumen, vasoconstriction just means contract that smooth muscle so the space in the middle gets smaller, right? You're basically just making a narrower pipe. That means more resistance, less flow, right? And of course, the opposite is true. If you vasodilate, that opens up the blood vessel, decreases resistance, and that would increase flow. And so these muscular arteries can control how much blood goes to each individual organ. For example, when you're exercising, you would vasoconstrict arteries to your GI organs and kidneys, and you would vasodilate to the muscles that are doing the contracting to make sure they get enough oxygen to carry out the workout. Right. And then lastly, arterioles. Arterioles also constrict and dilate, and they're just basically small arteries. This is an enormous part of what controls systemic resistance, right? And that's also sometimes called TPR, total peripheral resistance. So you'll use these arterioles to regulate blood pressure through the entire body. And that's the second part of this that we need to talk about, blood pressure. So I'm going to make a quick list, and we're going to title it Variables that Affect Mean Arterial Pressure. Mean arterial pressure just means average systemic blood pressure in your arteries. When you take a blood pressure cuff and you get a reading of 120 over 80, those are two separate numbers. If we take a time average of them, there's an equation that you can use to estimate the average. For this, it would be somewhere in the mid-90s-ish. That's the average over time. Okay, so variables that affect mean arterial pressure. One, cardiac output. right? And remember, cardiac output is heart rate times stroke volume. So if either your heart rate or your stroke volume change, your cardiac output changes. And the effect is pretty straightforward. An increase in cardiac output means you're going to get an increase in mean arterial pressure because you're pumping more blood out into the aorta. Makes sense. Conversely, of course, if you decrease cardiac output, you'll decrease pressure. All right. Thing two is we'll call this vascular diameter. And by that, I mean constricting and dilating, right? So vasoconstricting, right? That means that you're increasing resistance. And increased resistance leads to increased systemic pressure, right? Think of this as back pressure. Just like if you kink a garden hose, you'll get a jump in pressure behind the kink, even though pressure and flow go down after the kink, right? So this is causing back pressure towards the heart. We increase MAP. If you vasodilate, just the opposite. Decrease resistance, and that will decrease pressure. Now, just remember, you can do multiple constrictions and dilations at the same time, like my example. During exercise, you would vasoconstrict GI organs and kidneys, perhaps. You would vasodilate to the muscle that's contracting. And so in the end, you're just redirecting blood, right? Your cardiac output is going up, pumping more blood. You're vasoconstricting to the stomach and intestines and kidneys, and you're driving that blood to the muscles that need an enormous increase in blood flow to give them the oxygen that they need. All right. Last variable, blood volume. This is more of a kidney issue, so it'll be dealt more thoroughly with in the kidney section. For right now, just remember, an increased blood volume means increased pressure, and a decrease in blood volume means a decrease in pressure. The catch is you need to keep your blood volume at homeostasis, but we don't really, regulate this one up and down that much for the sake of pressure, right? These two are very easily adjustable, right? You can make your cardiac output go up and down. You can make blood vessels constrict and dilate. It's easy. It's reversible. Blood volume is not easily reversible. And so, you would never drop your blood volume to drop your pressure unless your blood volume is already too high and you just need to go back to normal. And so this is more of a stable homeostasis thing, at least it should be, right? These two are the ones that really get adjusted, right? And this is what your brain is keeping track of, right? So you end up getting this feedback loop where if your mean arterial pressure goes down, right, that is sensed by pressure receptors called baroreceptors, right? They live in your aortic arch and your carotid sinus, so in your major arteries. You'll have a decreased stretch of those baroreceptors. They're just pressure sensors in the wall of the artery that can tell how high the pressure is. And that signal gets sent to the CNS, and your brain says, oh, our pressure is too low. We should activate the sympathetic nervous system, right? And that will release some combination of epinephrine and norepinephrine. And epi and norepi have the effect of making the heart beat stronger and faster, so they increase cardiac output. They also have the effect of systemic vasoconstriction of arterioles, right? Both of those things will increase your mean arterial pressure to help us solve the problem, right? And so the point I'm trying to get at in all of this is there are two different reasons why you vasoconstrict or vasodilate. One reason is to change flow, right? We need more oxygen, more blood flow in a particular organ. The second reason is to adjust your pressure. And so it gets confusing. Sometimes you're vasodilating because you need more blood, and sometimes you're vasodilating because your pressure is too high and you need to go down across your entire body. They're both valid reasons to constrict or dilate. Okay, so in the end, what we're doing is, when I say a coordinated dilation and constriction of blood vessels, coordinated just meaning you're going to constrict to some organs at certain times and dilate to others, right? We're never constricting or dilating the aorta. That's all elastic. It doesn't even have much muscle to contract anyway. But all these other organs, livers and stomach and kidneys and muscles and whatever it might be. Any of these can either constrict or dilate as needed. So if you're digesting a meal, just relaxing, you would expect blood vessels to the GI organs to dilate. If you're exercising, you would expect them to constrict and the muscle arteries will dilate. That lets you shift blood where you need it, when you need it. All right. A few other quick things to wrap up about blood vessels. One is if you follow their pressure is always highest closest to the heart. So this graph is showing aorta, and then this is distance away from the heart as you keep moving further and further out. Notice we have a very steep drop when you go into arterioles and capillaries. It's just because they're small. Small vessels have a high resistance. That saps your pressure. By the time we get to the big veins in the vena cava, pressure gets really, really low. And the only reason that we can get away with that is those veins have incredibly low resistance, right? And that's just because their lumen is huge, right? And so arteries have slightly higher resistance, but they have high pressure to compensate. Veins have incredibly low resistance. That's how we can still get blood flow. Because remember, blood flow through the vena cava has to be exactly the same as the aorta for blood to come around in a full circle. So aorta is higher pressure, higher resistance. Vena cava is lower pressure, lower resistance. Right. The other important thing to see here, this graph showing cross-sectional surface area, just notice the vast majority of your surface area is in small vessels, right? Almost all capillaries. There's a reason for that, in that capillaries are the only blood vessels where gases and nutrients are being exchanged. So O2, CO2, glucose, they only cross capillary walls. The other vessels are just pipes. Their walls are too thick. You're not getting gas diffusion. That's the whole point of having capillaries be so tiny, is to increase their surface area to diffuse more gases throughout the body. And that's why the velocity of blood flow also slows down in the capillary, right? That slowing down is actually a good thing that gives us time for diffusion to take place. All right. Another interesting thing, veins. Veins aren't regulated nearly to the degree that arteries are. They do have smooth muscle. They can constrict and dilate. But a big part of what helps them move blood back to the heart is that veins actually have valves inside of them. These valves look very similar to what the aortic and pulmonary valves look like in the heart. And what happens is when muscles throughout your body, especially in the legs, contract, the muscles are squeezing on the blood vessel and they pressurize it. Since these veins have valves that close if blood tries to move backwards, but open if blood tries to move forwards, your skeletal muscles can actually squeeze that blood and push. And so the heart really isn't the only muscle that pumps blood. The muscles throughout your body, especially in the lower half of the body, because they're fighting gravity, they can squeeze veins, push blood back to the heart. That ends up increasing your EDV because it's pushing more blood and pressure into the vena cava, which fills the heart fuller during diastole. This is why walking during and after exercise is good for circulation, because if you sit down, your leg muscles aren't contracting and blood starts to pool in the lower body and doesn't get returned as easily. All right. Okay, last thing for blood vessels. This is showing movement and filtration in a capillary bed. And the idea is this is like an individual capillary. These would be endothelial cells lining that capillary. And the idea is fluid can leave through the capillary wall and nutrients can go along with it. And fluid and waste products can also come back in. So what you end up getting is filtration just means that on average, water leaves the capillary at the beginning of the capillary bed, right? So this would be an arterial over here leading in. This is on the way out to a vein. And remember, the further you get from the heart, pressure goes down, right? So on the arterial end, blood pressure is high, right? But we have to think about two different pressures. Hydrostatic pressure is just what you think of as normal blood pressure coming from the heart. Osmotic pressure is something that happens because there's an enormous amount of serum proteins in the blood, and those proteins are trapped in the blood and they can't diffuse out. That creates osmotic pressure to try to draw water back into the capillary, and it balances out hydrostatic a little bit. Okay, so on this end, hydrostatic wins. 35 hydrostatic pushing out, 25 osmotic pulling back in. So the net pressure of plus 10 means the hydrostatic pressure wins, and we get some fluid the capillary bed. Now, as we move down towards the other end, the venous end, hydrostatic pressure is going to drop, right? Eventually, it becomes lower than osmotic. And so now we only have 18 worth of hydrostatic pressure pushing this way, but osmotic pressure doesn't really change, so we still have our 25 pulling that way. That means that at the venous end, the osmotic pressure wins, right? And we pull some of that blood back in, or some of that fluid back into the blood. All right, now if you notice, the plus 10 over here is a bit bigger than the minus 7, and so there is a net loss of fluid into the tissue. If you didn't get rid of that fluid, it would just keep building up forever, right? And you would get edema and swelling. That's no good. And so what they're not showing you in this picture is that surrounding these blood vessels, these capillaries, are lymphatic ducts. And the lymphatic system is just a network of ducts that run through lymph nodes that carry excess fluid back toward the heart. All right, so any of that liquid that isn't pulled back into the capillary on the venous end of the capillary bed, that fluid is going to wind up in these lymphatic vessels, and they all dump back into the heart around the subclavian vein right before the vena cava. And so the fluid all works its way back to the heart. This just gives us the added benefit of passing through lymph nodes. Remember, most of your white blood cells aren't actually in the blood. They're in lymph nodes. And this way, if there's an infection, the virus or bacterium that's infecting you will get pushed through that lymphatic fluid and hopefully alert lymphocytes in the immune system that an infection is taking place. All right, as always, hope this was helpful. Follow along in the playlist, watch the videos in order. It'll make more sense that way if you can.